So I think there is a lot of confusion still about the whole PCR test protocol. Like, how does it work, and why are they not reliable in the first place? Because I often still hear people and they don't know. Could you, for those people, could you explain briefly, like, how does it work, a PCR test, and why is it not reliable?
Well, first of all, a PCR test is an a specific test. It's not a specific test for SARS-CoV-2. It's a specific, so it can trace any kind of viral load from other coronaviruses, for SARS-CoV-2, for influenza viruses. It can respond on many uh, viral parts. What does the test do? You take the genetical structure of the potential virus that they took from your swab, and it will be amplified for in between 40 and 50 cycles. So this means the genetic structure will be enlarged 40, on average 45 times. This gives you about 3 trillion genetic combinations. And within the genetic combinations, you can start searching for the viral part that might have infected you. The thing is, if you go over 27 to 30 cycles, you can find all infections. You will encounter all infections. If you had an infection with a coronavirus, let's say a year ago, it will still test positive. If you have been in contact with influenza, it will test positive, even if you don't have symptoms now. So what most labs in the world do is they use a 40 to 50 cycle rate. And then you can find anything. Even if I sneezed on a plant that is in my room a year ago, and you would take a swab of the plant and you put it into the machine, it will test positive. So there's the first thing what goes wrong. It has been published several times now that we can't exceed the 27 cycle thresholds.
Is there any place that actually does that, that uses the 27 or even below that?
That's a huge question. We don't know. We asked here at the local government to inform us about the labs and in which cycle thresholds they are doing the tests, and we didn't get a response. So some lab technicians took pictures with their mobile phones of the machines and sent it to us. Most of them were in between 42 and 48. So you get a lot of false positive results. So if we would follow science, which we know now, and they did blood. It's also a paper, I can't remember, it was Nature or The Lancet that published a huge article in which they took serological testing, blood samples, which are most accurate. And then they did the same, in the same population, they tested with a PCR test. And so what did they saw? If you surpass a 27 cycle thresholds, you get an enormous amount of false positive results. So the conclusion was, if you pass 27 and you see a positive result, you should combine it with a serological test as well to double check if it is a positive result. But most labs still use above 35, 40, 45 cycle thresholds.
Listen to the entire episode of the Immunity Podcast.
Link in the description below.